Welcome to ScienceMed Whiz. Today we are going to talk about transmission of genetic information from gene to protein. The agenda for today is nucleic acid function, nucleotides and nucleosides, sugar phosphate backbone, pyridine, purine resources, and the closing quiz. So nucleic acid is one of the major classes of organic biomolecules, the rest being carbohydrates, lipid, and protein. Then we go on to the monomers. Monomers are subunits, which are called nucleotides. These are three-part structures consisting of nitrogenous base, a pentose sugar, and a phosphate group. There's also two types of nucleic acid, which is the DNA and the RNA. DNA consists of two polynucleotide strands that are bonded together in a double helix formation, as you can see here in the picture. Then we have RNA. RNA is a single-stranded but much shorter in length. Its nucleotides consist of the sugar ribose, and it has all the bases found in DNA with exception of thymine. In DNA, it is consisted of polynucleotide strands, which are bonded at a double helix formation. So this means nucleotides found in the DNA contain pentose sugar 2-deoxyribose, and there are four possible nitrogenous bases, which are adenine, guanine, cytosine, and thymine. In RNA, there's an exception of thymine. Instead, it is replaced with uracil. RNA is made up of using a DNA template and is directly involved in synthesis of protein. So in this next picture, we have a better image that explain the nucleic bases of RNA and the DNA. Then we have RNA here, and we can see here it's a single-stranded, and then we have the double-stranded, which is DNA. We can also see the cytosine, guanine, adenine, and uracil. And then we can see in the DNA, we see cytosine, guanine, adenine, thymine. And you can see the differences between each. Uracil is where the RNA is, and thymine is where the DNA is. We can also see the helix of sugar phosphates between these two strands, and between also the single strand. We can also see the base pairs in the DNA, and then we can also see the nucleic bases in the RNA. So we're going to go on to and talk about nucleotides and nucleosides. They're both kind of similar, spelled, but they are not the same. Nucleotides and nucleosides. The difference is they end T and S, but they have different definitions. So nucleotides and nucleosides. Nucleosides are composed of a nitrogenous base, which is adenine, thymine, guanine, cytosine, or uracil, which are bonded all together with a pentose sugar by the glycosidic linkage. And I'm going to show you a picture in the next slide that we're going to talk about the glycosidic linkage. Then we have the nucleotide, which is formed with one or more phosphate groups that are linked all together by a sugar component of nucleosides. The bond between the sugar and the phosphate on the nucleotide is an ester linkage. So the differences between each, I want everyone to note this, is that nucleotides are formed with one or more phosphate groups, and they are linked to a sugar component of nucleosides bonded between the sugar phosphate on nucleotide, which is an ester linkage. Now, compared to nucleosides are composed of a nitrogenous base, which is adenine, thymine, guanine, cytosine, or uracil, bonded to a pentose sugar by glycosidic linkage. So on nucleosides, it's glycosidic linkage, and in nucleotides is ester linkage. Want everyone to point that out once they're in class, or they say, oh, what's the differences between nucleosides and nucleotides? Simple, right? Nucleosides is composed of nitrogenous base. You list the nitrogenous bases bonded to a pentose sugar by a glycosidic linkage. Then we have nucleotides, which are one or more phosphate groups linked to a sugar component of nucleosides. They're bonded between the sugar and phosphate nucleotide, which is a component of an ester linkage. And the bond between any additional phosphate groups is anhydrosis bonds. Okay, so also right here, I want to mention the nucleosides containing ribose are also called ribonucleosides. Okay, while those that contain deoxyribose are called deoxyribonucleosides. And nucleosides are synthesized from smaller molecules in the cell, but they are usually the result of breakdown of nucleic acid, which are the polymers of nucleotides in the diet. Okay, so nucleotides, right? Let's go back to nucleotides because I want to mention that nucleotides with 1 to 2 to 3 phosphate groups is called nucleoside monophosphate, nucleoside diphosphate, and nucleoside triphosphate, both all respectively, right? Nucleotides that are incorporated into that chain only have phosphate groups. So in this next picture, we are going to see that here, right? So we have nucleoside triphosphate. So where is this located in the structure? So this is located right here, okay? Right there, it is located all the way up here, right? This is nucleoside triphosphate. Then we have here incorporated nucleoside diphosphate. Here we have monophosphate nucleoside. And up here, we have the molecule pentose, right? Pentose. Why is it pentose? Because there's 5 carbons, right? Pentose 5. So here is the nucleus, the nucleotide structure, right here. Here we can see the glycosidic bond. Here we can see the OH, ribose, and the oxyribose, right? And the hydrogen and OH. Okay, and then we also see the 3 to 2 bond. So now let's go on to the sugar phosphate backbone. Sugar phosphate backbone. Here we're going to look at the nucleic acid and the chain of nucleotides that are held together by strong covalent bonds. Please make note of strong covalent bonds, known as phosphodiesters. This word is going to come up multiple times in your exams. Want you guys to make note of why these are held together by a strong covalent bond known as phosphodiester bonds. These bonds involve the phosphate groups and sugars of nucleotides, but not the bases, okay? So here I'm gonna elaborate more on this sugar phosphate backbone. And I'm going to talk about in a nucleotide, an ester linkage exists, right? So between the phosphate group, the 5-carbon of sugar, when the nucleotide is incorporated into that chain, it forms a second ester bond to the 3-carbon of sugar on the adjacent molecule, on the adjacent nucleotide. I'm going to say that again, but I'm going to explain it more with a picture because I want you guys to get an image here, right? So like I said, when the nucleotide is incorporated into the chain, it forms a second ester bond to the 3-carbon of the sugar on the adjacent nucleotide. We can see it right here, actually. We can see the 3-carbon of the sugar, and then we can also see the 5 that I mentioned earlier about the carbon and the linkage existing between the phosphate group at the 5-carbon of the sugar. So any additional phosphate groups in the unincorporated nucleotide, they are seen in the nucleoside diphosphate and nucleoside triphosphate, okay? These two different, so they are seen, make note, in the nucleoside diphosphate and nucleoside triphosphate. And then they are removed before they are added to a growing polynucleotide. The repeated phosphate-sugar sequence is the backbone of nucleoacids. Okay, please make note of that, what I'm saying. It's very, very important. So then we're going to go on to the pyridine and purine residues. So what is pyridine? Pyridine has a single carbon nitrogen ring, while purine has a double ring. So I'm going to elaborate more on this and the types of these nitrogenous bases that make up the nucleotides found in nucleic acid, right? So pyridine, that single carbon nitrogen ring, right? It has one single carbon nitrogen ring. Please note that. And pyridine has a double ring. More specifically, purine consists of pyridine that is infused in the imidazole ring. Okay, so more specifically, there are 3 pyridines found in nucleic acid, which are the thymine, which is only in the DNA, uracil in the RNA only, cytosine, which is the both DNA and RNA, and then we have the purines, which are only found in the DNA and RNA, and are adenine and guanine. Okay, please make note of that. I'm going to re-say it again because I want you guys to write this down in your notebooks. Pyridine, okay, maybe I can actually write it here. I can do a box, a table that is going to explain it a bit better. So we have pyridine, and then we have right, we have these two. This is double double ring, and this is single ring. Then we have adenine. I'm just going to put an A for adenine, guanine, G, C, cytosine, T, thymine, and U, uracil. So here we're going to describe each. So black. Okay, so adenine is in DNA and RNA. Guanine is in DNA and RNA. C, which is cytosine, DNA and RNA. Thymine is in DNA only. Only. I use the word only because it's only used there. No exceptions. Uracil is in RNA only. Okay, so this is a great table to copy in your notebooks to remember where the pyridine and pyridine go into. So conclusion quiz. So I'm not going to go over this because I want you guys to do it, yourself in your notebook. So these are specific questions that I went over. What are nucleic acid? What is DNA and RNA? And what are their differences? Nucleotides and nucleosides definitions. Nucleotides with 1, 2, 3 phosphate groups. And what are pyridine and purines? I just went over all these questions. These are all condensed into where you can write your notes under each question. Or as you went on into the video, you can pause this and go back in your notes and answer them. So if you have any other questions, please contact me. This is also coming from the MCAT Mometrix book. If you guys are in medical school, or you are a STEM student, or you are a biology student, chemistry student, bioengineering, all these other STEM fields, please, this is very important to write these notes and to keep in mind because you're going to use them for your state exams or even for med school because this is going to keep coming over and over again. So please review it. Have any questions, please text me, email me, and I will do another video explaining more about the MCAT. And don't forget to subscribe and hit the bell icon to watch new videos when they come out. Thank you all, and have a great day, everyone.